Reverse transcription is synthesis of DNA complementary to RNA, or cDNA, and is involved in many applications from gene expression to RNA sequencing. Reverse transcription, using in vitrogen SuperScript IV Reverse Transcriptase, allows favorable binding and speed for better cDNA synthesis. SuperScript Reverse Transcriptases have been a trusted product line with more than 50,000 publications and references. The speed of cDNA synthesis is related to processivity of the reverse transcriptase, which measures the number of nucleotides added in a single binding event. Highly processive enzymes like SuperScript IV Reverse Transcriptase enable synthesis of longer cDNA in as little as 10 minutes. With all the reaction components pre-mixed in one tube, in vitrogen SuperScript IV VILO Master Mix also helps to ensure high data reproducibility and avoid contamination from repeat pipetting. With high efficiency, SuperScript IV VILO Master Mix yields more cDNA and thus lower CT values in RT-qPCR. This enables detection of low RNA input from limited sources. For more information about how SuperScript IV Reverse Transcriptase can facilitate success in your molecular biology research, please visit thermofisher.com/4vilo.